Good morning, students. Buenos días, estudiantes. 早上好，同学们. Bonel du matin. Salamat pagi, morid morid. Guten Morgen, studenten. Thank you for joining us today, and we want to continue with our video presentations for the community health modules. And just to recap, there's a lot in the news about community health these days. And so far, we have had video reviews of the global virus transmission and community transmissions, and how there are steps taken through social isolation and others to prevent the spread of the disease within the communities. We have also looked at community health care and COVID-19, and we looked at adding resources and capacity to the healthcare system to have effective treatment for people who have been infected with the virus. We looked at COVID-19 symptoms and complications called the sequelae. And there's been a lot of talk about findings for different sequelae and different complications in the news, and it's worth looking at. Today, we want to look at how to make a vaccine, and we'll focus a bit on what work that's being done to make vaccines for COVID-19. We'll discuss some of the science, the discovery, the development, the production processes, and some of the proposed changes to the process to speed up the benefits of the science. So when we get started on this today, there are going to be a need for researching some of the vocabulary that you're going to hear. And some of it has to do with the science, some of it has to do with the organization of the development and production of the vaccines. Something to look up, the Coalition for Epidemic Preparedness and Innovation. That has been formed to look at collaboration between countries for the development of vaccines against the new high-risk viruses that are anticipated. And right now, the coronaviruses are causing havoc. COVID-19 is not the first coronavirus. SARS, MERS are viruses that you may have heard of in the news in the past. The coronavirus that we're dealing with now, COVID-19, is actually termed as SARS-CoV-2. So it's within the family of the SARS and MERS, but it is different. Now there's an urgency to find an effective vaccine for COVID-19. And COVID-19 seems to be the worst of all. Other terms that we're going to look at are such as disease enhancement, fully sequenced genome, covalent, nucleotide sequence, RNA, and cGMP. cGMP stands for current good manufacturing practices, and that's one of the major steps that is done in the production of vaccines. This COVID-19 is called a novel virus, meaning that it hasn't been seen before. There's a need to put in place processes that are going to be effective for developing vaccines and treatments against this. I wanted to draw our attention to what we call the routine flu vaccines. Flu vaccines have been developed and in use for a period of time, and there is a process that incorporates global cooperation so that the vaccine can be adjusted to the viral mutations that occur on an annual basis or close to an annual basis. So COVID-19 is brand new. There's no infrastructure to study it, to prevent it, or to treat it at this time. So we're all learning real time. And if we want to draw from experience to accelerate our learning, then we can look at the developments for the influenza vaccine. A little history: in 1918, the influenza vaccine caused more deaths than the effect of all of the warfare in World War I. So it effectively caused the end of the war because countries didn't have any soldiers left they could send to the front. The vaccine that we're drawing for, the vaccine experience that we're drawing from, shows that there's cooperation 100 or more national vaccine and influenza centers around the world, and they're focusing on identifying the mutations in the laboratory and producing vaccines that can prevent the effect of that mutated virus every year. In producing vaccines, there is a traditional technology, and particularly with development of the COVID-19 vaccines, there's new technologies that are going to be used. The traditional technologies have been in place for about 70 years. It was found that large-scale production could be accomplished by injecting the virus into chicken hen eggs and allowing the virus to replicate over 7 days, extracting the fluid in the eggs in which the vaccine has been growing, and that's called an ovalbumin. Then in the laboratory, process is done to kill the virus in the fluid or to weaken it so that it is no longer infectious but still identifiable by the body. Then the vaccine is created, and that either dead or live attenuated vaccine is injected, and the body's response to those foreign pathogens is to create antibodies to defend against it. So it's harmless when it's injected, and the body recognizes the physical forms of it and defends against those. So when you are in contact with the real harmful virus, the body's already prepared and just says, yeah, we've seen this before and we know what to do to kill it. So in that process, typically it takes 1 egg for 1 vaccine. And use the influenza vaccine as an example. Billions of doses are produced each year. So that means that billions of eggs are used. This is a very complex logistics process, and it has to be very carefully managed. So many eggs, so many doses. And that process takes about 12 to 18 months to identify the potential mutations of the virus and to produce the new vaccines that will be effective against those mutations. And as I mentioned, in the body, what happens is that the non-active virus or the shells of the virus are introduced through the vaccine, and it triggers the body to produce its own antibodies to fight against it. Now we have a novel process and a novel virus. And what's been done is to look at the process for using messenger RNA vaccine production. This has been a process that's been used to make vaccines for animals, but it has not yet been used in humans. So in the lab, it's known what to do. The effect of the product that's made on a human is not yet known. So that's where the risk lies, is in getting this right the first time. And the first human trials are underway. I believe that the first participants may already have been enrolled and possibly have already been injected with the prototype vaccine. That messenger RNA vaccine was developed in 42 days versus the 12 to 18 months that's been the lead time needed to develop a vaccine using the traditional method. So you can think of the messenger RNA process as a training camp for the body. The vaccine tells the body what antibodies to produce so that they can attack the potential pathogens. Within the laboratory, the process for this is something that we have discussed in the biotechnology module in community health. And in that, the RNA strand is manipulated. So the process then is that this nucleotide sequence, the introns and exons, are replaced. So the introns are removed by RNA splicing, and the RNA matures, and the exons are covalently bonded to another RNA strand in order to create the mature messenger RNA. And this process codes the RNA with the message you want to send in the vaccine, which is telling the body to prepare defenses against the potential pathogen. Testing for vaccines is a time-consuming process, as it is for any pharmaceutical product. With any clinical trial, there is a prime operating principle that is first do no harm. So this process is developed to first look at safety and then efficacy and then the effectiveness over large populations. In the preclinical phase, where a lot of work has been done traditionally with animals, then the effect on animals can be measured and described. When it's deemed that you have something that could be effective, then it is brought forward to what's called a phase 1, and it's tried on healthy people just to make sure that it doesn't do any harm. It does not produce the disease enhancement whereby the vaccine causes the illness. In phase 2, the number of people involved in a trial would increase, and you're looking to see the safety and efficacy of the product. And again, looking at disease enhancement. You are not causing the symptoms to occur in people as a result of injecting them with the vaccine. Phase 3 is an additional safety and efficacy, but it's on significantly larger populations of thousands of people. And this can require 18 months. And it's also, as I mentioned, a very complicated logistics process to manage all of that. Once the vaccine has been improved, then the companies go on to develop the process to manufacture it in large quantities for distribution. That follows what's called the current good manufacturing process, which most companies or all companies know how to follow and to document. Then the FDA comes in and inspects to make sure that the companies are doing what they say they're doing and that the process is safe and the product that's manufactured is consistent batch to batch or lot to lot, as they say, and that it's effective when it's used in the community. A new method has been put together for this, and it's called an outbreak paradigm. And that's to get the product to market faster. Right now, we have an urgency to get a vaccine for COVID-19 onto the market. So to accomplish that, there has been a review of some of the steps that have been taken in developing malaria vaccines in which they're used in real time. And the objective from this outbreak paradigm or human challenge trial is to speed the time to market by 6 to 12 months. Many steps are done in parallel instead of undertaking them sequentially one by one. So the discovery, development, trial, and manufacturing investments are undertaken at the same time. As an example, in the United Kingdom, the National Health Service has decided that they will place an order for 1 million doses of a vaccine that they have never even seen. And that pays the manufacturers and the scientists to do the work in the hope they can get something that will be effective. If it's not effective, it's thrown away. It's already bought and paid for. And as I mentioned, the objective is to speed up the process. We see the rate of transmission of the infection in the communities around the world. So if we can interrupt that by a few months or even a few weeks, it would be an incredible benefit to the country and to the world. So I want to thank you all for taking time today to look at this presentation. Email me if you have any questions. Email me if you need any assistance. Keep safe, keep strong, keep healthy, and keep studying. And remember, wear a mask when you're in public to protect those around you and to protect yourselves. Thank you.